Okay, in this video, we're just gonna pick up where we left off with the last one, and we're gonna wrap up these last four organelles. So the next couple of slides just look at the organelles that we've already reviewed and just giving you some information about their structure, which is not so important to us, but it's there if you want to look at that. And if you look at the function, you'll see that this really overlaps with the stuff that I included in previous slides. So take some time to look at that slide if you want to, or at these slides if you'd like to. So let's jump right ahead into the cytoskeleton. The cytoskeleton is pretty much exactly what it sounds like. That root cyto, C-Y-T-O, means cell. It refers to the cell. And skeleton, well, you know what a skeleton is. And so this is really the skeleton of the cell. And so what it does is, much like our skeleton, it helps maintain shape of the cell. And so, you know, we're going to get into bones later for our course, but this really is the skeleton of the cell, and it's made up of a whole bunch of different protein fibers. So here, maintain shape and internal organization. So the three protein fiber types, or filament types, we're going to refer to these as filament, which is just a protein fiber in this context. And these are microfilament, intermediate filament, and microtubules. And so these go from smallest to largest. And so when we look at these, you can see that these are microtubules, and these are more microtubules. And it says here, the motor proteins attached here. So what the cytoskeleton allows, not only does it give it general shape and form to the cell, but it actually serves as kind of a roadway within the cell. And so what that means is, let's say right over here where I'm highlighting, let's say I make some protein right over here, and I want to shuttle it off to this part of the cell. So in the old days, nobody really knew how within the cell, intracellular transport actually happened. It was almost like, well, is it just a watery goop in there? We cross our fingers and hope that this product makes its way to that other end of the cell. But then what people discovered was that this cytoskeleton actually serves as kind of a highway. And so what you'll see is that within the cytoskeleton, that's why I have this term motor proteins here, we can actually hook up or latch on some of the things that we end up making here, and it hooks up to the cytoskeleton, and through a motor protein makes its way from point A to point B. And that's what this is showing you. So you see that we've got an ATP powered, this motor protein, see, motor molecule, ATP powered, and some organelle or some substrate that I've synthesized within your cell. It goes from point A to point B because that ATP molecule fires it up, and it almost just seems to kind of crawl along this cytoskeleton framework, these microtubule frameworks. And so in any case, the cytoskeleton serves as an intracellular road. So, you know, I keep saying that term, and I don't know that at any time I've really been explicit about it, but it's worth your while understanding the difference between intercellular versus intracellular. And I'll just type those words up there, cellular. And so intercellular means something that happens between cells. For example, we talked about intercellular junctions or things that connect two different cells together. Intra means within. And so for those of you who are sports fans, maybe you've heard of people talking about intramural sports, which is sports, you know, within just one organization, maybe intramural sports at some college, for example, or inter-league play, so play between two separate leagues or parts of a league. So anyway, inter and intracellular are words that you should be pretty comfortable with. And so cytoskeleton allows for intracellular transport. Okay. Now the next two are really kind of offshoots. Cilia and flagellum are really kind of offshoots of the cytoskeleton because it's made up of the same or similar types of protein components, but they're a little bit different. And so what ends up happening is that these are projections of the cytoskeleton. The first is a cilia. And so if you look at here, what we have here is, let's see, 1, 2, 3, 4, 5, 6 different cells just laid up over here. And sticking out of the cells are these little pipe-looking things, these little blue pipes. And what these little blue pipes do is that they allow things to move along the cell surface. And so this is a pretty famous picture. This, maybe I shouldn't, I don't think that's the right way to say it. It's not a famous picture. It's a pretty commonly used picture when we describe the function of cilia. So this is something like in your airways, like let's say your air pipe, your trachea. And so in your air pipe, there's mucus that is lining that pipe, and there's cells. And so gunk and different things in the air get stuck in the mucus, and then these cilia will sweep those things, this sweeping motion, and this is showing you this so-called power stroke and recovery stroke. You don't need to know those terms, but it's almost like if you ever swept anything with a broom, like a traditional broom, you sweep it left to right, and then you reset and sweep it left to right, reset, and so on and so forth. This is what's happening with these cilia. So what is the function here? It is to move stuff along the surface of a cell. That's what cilia do. And these are really considered to be projections of that cytoskeleton, again, made up of things like microtubules, again, so very similar constitution. So that's what cilia do. So if that's the case, if that's what cilia do, then what do flagellum do? And so flagellum or flagella as a plural. So what those do, we actually only see flagellum on one cell type, and that's in sperm. And what this is the cell, the main part of the sperm, and this is the flagellum that sticks out, and that allows the cell itself to swim. And again, really, the only example we have for human physiology is in sperm cells, but flagellum are found in other types of microorganisms and such. And so, really, by definition, what that means then is that a cilia will move things along the cell surface. So the cell itself doesn't move; it moves things around it. But the flagellum allows the cell to move, and that's to swim forward. Okay, the next thing is a centrosome with centrioles inside of them. And so probably the best way I can describe centrosomes and centrioles is this. I have an image here, and if you take a shelled peanut, the whole thing is the peanut, but when you crack that thing open, inside of it are two peanuts. I don't know if there's a better word in the food industry to talk about that, but you know what I mean. Is that the larger entire entity is the peanut, but then within it are the actual peanuts that you eat. And so similarly, a centrosome is like the larger shelled peanut, but on the inside, the parts that really matter are the centrioles, and those are like the little edible portions inside. So there are two centrioles within a centrosome. So a centrosome, by definition, is a microtubule organizing center, and it contains two centrioles. So the centrioles themselves, we see them at the base of the cilia and the flagella that we just described. We actually see the centrioles position right over there. And I didn't point it out at the time, but there it is right there. There's a picture right at the tip of that cilia, and you're going to see a centriole there. But you'll also see that in flagellum, although I don't have a specific picture for that. But probably the more famous function, if you could only say one thing about them, is this. They guide the movement of DNA during cell division. And so I don't want to go too much into that right now, because we're going to have a whole section just on mitosis and cell division. And there we're going to see the activity of the centrioles. But really, ultimately, what's going to end up happening is that when you take one cell and you divide it into two cells, that happens during mitosis, we need to evenly divide out the DNA. Again, which I'll talk about later, but then the DNA itself needs to move from one end of a cell to another end of the cell so I can pinch it off to form two cells. And the thing that guides the movement or the migration of that DNA are these things. And really, that is their most famous function. So that's what centrioles and centrosomes are. Okay, a centrosome has two centrioles, larger shelled peanut with the actual edible portion inside. Okay, and the last thing that we need to cover here is the microvilli. So the microvilli, again, we're only going to see in certain parts, and it's probably most famously found in the small intestine. And what you'll see here is within that cell, the cell membrane itself starts folding in, starts having these folds on them. So this, look, if you were to squint and look at this thing, you might be tempted to call those cilia. Okay, so if you look at this thing, these little parts that I'm highlighting, if you were to squint, you might say, when I squint, it looks a lot like these cilia that I just saw over here, but they are different. Remember that the cilia themselves are proteins, microtubules that stick out of the cell. But what these microvilli are, the microvilli are actually just the cell membrane, the phospholipid bilayer, folded up on itself. So they're quite different. And so the microvilli, what is the purpose there? Is the folded part of the plasma membrane to increase surface area. That will become more meaningful when we actually see it in the context of its appropriate organ system, like I said, probably in the small intestine. But what ends up happening is that, let's say I've got a bunch of stuff out here and it wants to diffuse on to the inside of the cell. Well, the greater the surface area you have, the greater the rate of diffusion. One more time. The greater surface area you have, the greater the rate of diffusion. So meaning the faster you can get this stuff from the outside to the inside, provided the concentration gradient is already there. So increased surface area allows you to do that. I think I used this example before where, a bunch of people, if they're in a room and you only had one door 3 feet wide, then it would take a certain amount of time for 100 people in a room to diffuse or to move out to the hallway. But if you had a wider 6-foot door or a 15-foot door or a wider 30-foot door, if such things exist, the greater surface area you have, the greater the faster the rate you can have for movement of those people from one room into the hallway, if we were to use that model of diffusion. So that's what microvilli are there for. So again, probably the most commonly confused thing among students here are microvilli and cilia. Those are probably the ones that students most commonly confuse. All right, so from this video as well as the previous one, we've now covered all of these organelles below. I won't go ahead and rehash them here, but that's all the relevant information you need to know for each of these organelles. On your PowerPoint, there's a handful of slides here that I'm not going to go over because we're going to cover this separately. This is on mitosis. It's just there if you want to see it, but I'm going to have a separate presentation just on mitosis, and that'll do it.